Okay, it looks like it's time to start. So welcome everybody. My name is Nancy. I'm going to turn on my video so you can see me. Hi there. And thanks for joining us today. So today we're very lucky to have Dr. Colin MacLeod with us, Curatorial Assistant for the Marine Invertebrate Collection here at the Beaty Biodiversity Museum. We're very excited to find out what he's been up to at home as well with his research. And so I'm going to just do a little introduction of the museum to start off with. So let's go ahead. Oops. And also joining us today is Kashifa from the another museum interpreter, and myself, Nancy. Thanks, great. And so where is the Beaty Biodiversity Museum? Of course, some of you have been there. We are on the ancestral, unceded, and traditional land of the Musqueam peoples, and we want to acknowledge that, and we love learning more about them. So if you'd like to learn more about them, we encourage you to visit their website at www.musqueam.bc.ca. And if you'd like to see it on the map, there it is, and we're right in the middle of UBC there, close to, for example, the Pacific Museum of Earth, kind of close to the bookstore, right in the middle there. There is what you would see when you arrive, when you get close to the building. And in the center there, you can see there's a skeleton. That is our blue whale skeleton. So this is our largest specimen, and it looks like that. And it is our biggest specimen of our 2.1 million specimens in the museum. So, and behind there, just behind the skeleton, you can see the Biodiversity Research Center as well. And together with the museum, we are the Beaty Biodiversity Center. So the research center is covering all sorts of biodiversity topics such as ecology and evolution and others. And if you went into the building, you'd go underneath the blue whale skeleton here, and then over down the ramp to the collections with the different rows. And so what's really unique about our museum is that over 95% of our collections are in the room with the museum exhibits and with the public and everybody else. So this is what it would look like. There's a variety of things, it's not just the big ones, it's all the small ones as well that are very important. And they are divided into 6 different collections. And so we're going to be focusing here on the Marine Invertebrate Collection today. And this is where it is on the map. And I'm going to let Colin take over and go ahead and start with his presentation.
Fantastic. Thanks very much, Nancy.
Thanks.
Brilliant. Hello, everyone. It's fantastic to be talking to you all today. So I'm going to talk a little bit about mobile labs and backyard parasites, although I'm using a little bit of artistic license when I talk about backyard parasites, because really I'm talking ones that exist not necessarily in our backyard, but it's they're in the Burrard Inlet, which I think we kind of all think that is pretty close to being our backyard. This photograph is, the background here is a trematoid cyst that I photographed embedded in barnacle tissue, and I'll have a few more photographs of this kind of thing as I go on. I've been working with parasites, which are definitely a subcategory of marine invertebrates for about 10 years now. And I'm lucky enough to have one foot in the Beaty Biodiversity Museum, taking care of the Marine Invertebrate Collection, and also in the Biodiversity Research Center as a postdoctoral fellow looking at parasites. So I'm going to talk a little bit about mobile labs. Actually, I'm going to talk mainly about my home lab during lockdown, but I thought I'd give you a little bit of a story about the fact this is my first time creating a lab for kind of mobile purposes. Back in 2017, I decided that I wanted to do a little bit of field work, but I don't have a car or a driver's license. So I thought, I'm just going to take my bike out and cycle to Ladysmith on Vancouver Island. Obviously, all the way I did take a ferry in there. So in this photograph, you can see the top right panel, that is my trailer packed full of all sorts of goodies, scientific equipment, my laptop, microscope, camera to attach to the microscope, obviously all my camping gear. And then the bottom right photograph is a little bit of a pause that I took on Lionsgate Bridge with my bike and my bag and my trailer. And this was mainly to get my breath back, because I just cycled all the way through rush hour traffic in Vancouver in the morning. And this was actually the first time that I'd ever taken a trailer attached to a bike. And I was a little bit over ambitious in how much gear I could put into it, so it was a lot heavier than I expected. Also, I will draw your attention to the fact that this journey did not take me 5 hours and 17 minutes. It took substantially longer than that. I think Google was maybe using a fitter version as a reference point. So at the other end of the journey, I found this beautiful little estuary just outside Ladysmith, and I started collecting snails. And that was the purpose of the journey, was to find new areas that maybe hadn't been looked at before, try and document what parasites were there, and almost invariably the hosts, the parasites that I work with, are marine invertebrates. So there's nice connections there with both my research and my work at the museum. These are more photographs from the trip. This is me in the bottom right-hand corner, looking a little bit wild-eyed after finally making it to the campsite after my very long mega cycle. The bottom left-hand photograph, that was after unpacking all the enormous amount of gear from my trailer. The top right photograph is the camera, the dissecting microscope that I took with me, and then a photograph in the top left-hand panel that I took through that microscope when I was very happy to find parasites in an invasive species of snail in the estuary. And the photograph in the middle is just my little setup inside my tent. You'll notice I've gone from full beard to braided beard, and that was because I had a little bit of a close call with the camping stove. So enough about that. This is my home lab during lockdown. This is actually the main photograph on the left. That would be essentially facing me normally. I would be sitting on the far side of that table, but just because of the lighting of my house, I thought it would be nicer to look at it this way. So on the left, I've got a compound microscope, which is allows me to take very close-up photographs of tissue specimens. And then on the right-hand side, there's a dissecting microscope, which allows us to use tools to manipulate pieces of tissue before transferring them to slides that I'll look at higher magnification. And there's a lot of other cool toys down there. I have the silvery object at the bottom left of this image. That's a counter, so I can count a number of different objects without trying to keep track of 3 different strings of number in my head. There's scissors and syringes and forceps and scalpels. And in the bottom left-hand corner, there's these sample vials so I can preserve tissue for future analysis. Of course, sometimes I don't have the tools that I need, and these 2 photographs on the right, those are 2 essentially sort of micro tools that I made out of. The top one is a thumbtack, and the bottom one is a sewing needle, and I sharpen them with a whetstone so I can make essentially tiny little scalpels to further dissect parasites that I had previously taken out of hosts. So today I'm going to talk a little bit about 2 different host parasite systems that are found in Burrard Inlet, and you would never know that they're there because really we just see the marine invertebrate that holds the parasites. So I'm going to work clockwise from Litterina and Cryptic. So the top left-hand panel, we have the periwinkle. It's native to BC. It has a huge range, and you've probably seen these. They're actually very small. You can see this from the scale bar on the right, but they're usually between maximum size about 1.5 centimeters from the tip scale. Inside the snail, we have this parasite, Marine. So top row center panel, that would be this kind of large sac-like object that lives inside the snail and produces these free-swimming infective stages known as cercaria. They're kind of a type of larvae. And the photograph in the top row right-hand panel, that is the cercaria. And so periodically, the cercaria emerge from the infected snail, they swim around, and they find the next host in their life cycle, in this case, Hemigraptus aragonensis, which is a common green shore crab, which I'm sure most of us have seen down at Burrard Inlet. And so the larvae goes inside the crab and forms these metacercarial cysts, or just cysts, and these are in the bottom row, 2 panels on the right-hand side. Now, I have the species of the parasite as Marine, but sometimes when you dissect marine invertebrates, you find surprises. And one of the surprises I found was these 2 cysts look really different. The one on the bottom right-hand corner has a really thick cyst wall, has quite distinctive brownish color, and you can see these well-defined dark shapes inside it. But the one on the left, again, a lot thinner external cyst wall, and it's just basically looks different in almost every way I can think of. So these are the kind of surprises that I really like finding, because it means that potentially I'm looking at 2 different species of parasite, but both infect Hemigraptus aragonensis as a host.
Hi, Colin. I just wanted to say that some people are having a little trouble hearing, so maybe if we slow down a little bit, that might help. It is just the audio is going a little bit like, yeah, a little smooshed together, I guess, is how I would describe it. So thanks.
Absolutely.
Or if you can maybe even speak a little louder if you can. Thank you.
I can do, I'll try and do both those things.
Okay, thank you.
I do tend to get a little bit carried away and excited when I'm talking about parasites. So it's possible that I'm just getting a little bit too fast, even for my computer to keep up.
Thanks.
And so for those people that may have missed something about the slide that's on the screen at the moment, this is the first parasite's life cycle I looked at. Litterina scuculata, a common periwinkle, is the first host in this life cycle. Inside that snail, you have these developing stages of parasites that develop into a free-swimming stage known as a cercaria. They swim out of the snail and then find a crab and then form these cysts inside the crab. And actually, I'm glad I got the chance to revisit this slide, because what happens next is that usually a seagull will come in, eat the crab, and then as the crab is digested, these cysts in the bottom row on the far right will burst open and an adult parasite will emerge, take up residence usually in the gut of the bird. They then mate and produce eggs, which exit the seabird rather glamorously in its poo. And then the eggs transform into a new stage, which then finds and infects the periwinkle, and that's our complete life cycle. Actually very neat. So the next parasite system I wanted to talk about, again, it uses really common species in Burrard Inlet. We have, unfortunately, Litterina scuculata has a little bit of a hard time. It definitely is infected by a large range of parasites, but hopefully it doesn't negatively affect the population too much. So again, we're going to work clockwise from the top left-hand panel. There's our snail. Inside the snail, you get these sac-like objects that are actually called sporocysts, and they produce, again, this free-swimming larvae called cercaria. This parasite species is Litterina gratiosum. Has a slightly different looking cercaria, a slightly shorter tail, and a slightly less pointy front end. And so this larval stage will emerge from the snail, it will swim around, and it will find the common barnacle, Balanus glandula. So I'm sure you've probably seen Balanus glandula around Burrard Inlet, or in fact in many rocky intertidal areas. It's very common, lives in really high densities. It settles on any hard substrate, whether it's rock or a mussel shell, as you can see here. And I usually collect barnacles that grow on mussel shells, really just because it's logistically easier rather than picking up a bag full of rocks with barnacles encrusted on them. I can just grab a handful of mussels and then take them back to the lab and then freeze them and dissect them. So in this case, again, we see the cercaria going into the barnacles, and this is really interesting. The barnacles feed by using these feathery structures that kind of comb the sea, and they pull out little particles of food, which the barnacle then moves through to its stomach and then it digests them. But unfortunately for the barnacle, sometimes it will catch one of these larvae, which will then get moved into the gut of the barnacle, and then they'll form these cysts again. And that bottom row center photograph, that's actually the smallest barnacle I could find that was infected by this Litterina gratiosum. It's actually 2 millimeters from end to end, and is so young that it only has one cyst in it. This is actually quite a surprise, or actually not, because it is so young, but I've found barnacles that have up to 150 or 200 cysts in them, although they are much larger. And then in the bottom right-hand photograph, these are another image of these metacercarial cysts embedded in the barnacle tissue. And you can see why I would absolutely love to spend time taking photographs of these, because they have a certain beauty to them. So I appreciate the barnacles not only because I appreciate the parasites, sorry, not only because they infect these very common species that we're all quite used to seeing in the intertidal zone, but understanding what's going on inside those organisms, I think, is absolutely fascinating. And I will leave you with an entire slide of what I have now started calling para art, where I get a little bit carried away, or maybe I'll need a little bit of a break from my research. So I start taking photographs of these cysts. I think the top left one is my favorite because I think it kind of looks a little bit like the dinosaur eyes staring into the car in Jurassic Park. And I posted that on my Twitter account, and people do seem to agree with me. That's the end of my presentation, and I'll be very happy to answer any questions that anyone has about marine invertebrates or parasites.
Oh, fantastic. I'm going to read one from Jeffrey, okay? So here is Jeffrey's question. He says, obviously, most crabs are not eaten by birds, so does the parasite not survive otherwise? Also, do any of these host species suffer negative effects?
Those are really, really good questions. So pretty much the parasites play a kind of numbers game. They know that there will probably be a small proportion of the cysts that they produce that will end up getting into a bird. So the number of those cercarial larvae that come out of the snails, those are huge. They can actually come out about 1,000 or 2,000 at a time. So if you imagine at each step, there's going to be a reduction in the number of successful infections. So the first step, maybe 50% of the cercaria find a barnacle and then infect that barnacle. And then a smaller percentage, again, will be eaten by the definitive host. Same goes for the crab. So a proportion of the cercaria will successfully infect a crab. But then you're right, not all crabs are eaten by seagulls. So it's really a kind of, the parasite tries to increase the probability of reaching that final host, and that's the shorebird. And actually that kind of ties nicely to your question about, are there any negative effects on the crab or the barnacle? Because sometimes those cysts interfere with, for example, a predator evasion response. So this is actually something I'd be really interested in researching in the coming months. There's a possibility that the cysts that form inside the crab, because they're almost all in muscle tissue. So there's a possibility that the cyst will interfere with the functioning of the muscle tissue. And that really just translates into the crabs are less able to run away from seagulls, or they're less able to hide and squeeze into small spaces. So not only is there quite often a negative effect on the host, like the crab or the barnacle, that negative effect is usually has evolved over time to maximize the probability that that cyst will get into that bird.
Great. I'll go on to Kashifa's question. Is there a reason why barnacles would grow on rocks versus mussels or other shells? And does the substrate a barnacle grows on affect the parasites they have?
Those are brilliant questions. As long as the substrate is hard and in a way semi-permanent, that's a successful settlement spot for barnacles. I mean, like I said, you sometimes see at certain times of year, actually, usually in, I think, February and March-ish, there's this huge burst or sort of reproductive season of the barnacles. So they jettison all these larvae, they come out in probably billions in Broad Inlet. And so they try and find settlement spots, but sometimes there's none left. So they'll maybe find that they settle on something like seaweed, and because seaweed will not live as long as the barnacle, then that's kind of just an unsuccessful place to sit or to settle. Most of the ones I've seen are on like boulders or mussels, and the mussels themselves, they attach themselves to the rock, so it's kind of an indirect permanent home for them. I found some barnacles that are growing on hard-packed sand, but that's not quite as good because a storm could come along and wash them away. And yeah, the second part of your question actually gets to something that I was really interested in, because if these little larvae come out of snails and they're swimming around and they're going to get washed up and down by the tide coming in and out, and then they will be washed over barnacles and they'll be washed over mussels. So I actually tested this and I thought, well, surely if you're a filter feeder and you're living on top of another filter feeder, there's a possibility that you'll have a lower number of parasites in you because they'll get sucked into the mussel before they get to the barnacle. And that actually turned out to be true. There was a significant reduction in the number of cysts that I found in barnacles that were growing on top of mussels versus barnacles that I was finding growing on rock. So yeah, and it's again, that's one of those, I really like that because it makes a lot of sense. There's no complex mechanisms. You just think, well, you've got two filter feeders. If you have two, one living on the other rather than one living on rock, then that makes sense that you would have a lower infection.
Great. And then we have a question about the study site. So why collect from Vancouver Island specifically?
Um, Probably a couple of reasons. I had heard that this invasive mud snail called Basilaria atramantaria had become established in a number of places along the mainland BC coast and around Vancouver Island. They came from Japan because people were shipping oysters from Japan to BC, and to stop the oysters from drying out, they would pack seaweed around them, and they inadvertently packed some snails along with the seaweed, and so you have these little populations of the invasive snail anywhere where there was somebody's tried to start up an oyster aquaculture business. And so I was interested to see if the infection levels in this introduced snail were as high as the infection levels in their home range in Japan. And so that was the kind of, I guess, the research reason for going to Vancouver Island. But as I've found out since then, there are plenty of barnacles much closer to home. So probably the secondary reason I chose Ladysmith was I thought I'd be able to cycle there in one day without injuring myself, because like I said, this was the first time I ever took a trailer, and I'm very glad I didn't decide to go further. But also Ladysmith seemed like a nice sized town. They had a nice camping ground, so it was a number of reasons just made it the ideal spot to try.
Great. Thank you. Okay, so Maggie, who loves birds, she asks, for the first parasite, the snail and crab are specific, but can it be any bird?
Yeah, so all these are fantastic questions. So there's different levels of specificity throughout the parasite's life cycle. And for the most part, the one stage that is most specific, or obligate we call it, are the snails. So it's highly unlikely that Maritime gratiosum would be able to infect any other snail other than Litterina scutulata. I think it's just, it has to do with the way the parasites have to overcome an immune response from the host, and potentially there's a very specific immune response from snails, so they just have to limit their repertoire of invading skills to just one species. There's certainly a little bit more flexibility around the second intermediate host. So maybe there are a couple of species of crab that Maritime lyricula can infect. Actually, I found one Hemigrapsis nudus, which is related to Hemigrapsis oregonensis, and it was also infected with these cysts. And then I have a feeling that there may be even more flexibility in the final host, because really they just, the parasite will have evolved to be able to deal with a highly acidic environment of the host's gut. So there's a probability that we'll be able to survive in quite a wide range of bird hosts. However, it's really hard to find any kind of hard and fast rules that stick across the board in parasitology. It's the same as any biological discipline. Biology just loves to prevent you from coming up with one rule that will fit everyone. So there are some species of parasite that are actually more common because they have flexibility throughout their life cycle. And then there are some species that absolutely need these 3 individual species to complete their life cycle, and they'll maybe be a little bit more limited in how far they can spread.
Great. Got a thumbs up and a clap for the not being able to come up with a hard and fast rule. That's good. That's from Mira. Okay, so we've got Melanie. Oh, sorry, Sheila. Let's just go to the. Sheila says, "Do your parasites generally follow a multi-host cycle?" Do your parasites, your
my personal parasites, yes, they do. I do. I think of trematodes as my personal parasites. I call myself, I mean, I am a parasitologist, but I have found more than enough within trematode parasites to keep me interested for, I mean, the last 10 years, and I'm assuming the rest of my research career. There's some variation within trematodes. Some of them have evolved to use 4 different hosts. Some of them have gone the other direction and they just use a single host. And that's kind of one of those really fascinating aspects of trematode biology, because you have to wonder, evolutionarily speaking, what is the benefit to more hosts, or what's the benefit to fewer hosts? And it must be specific to a particular habitat or specific to a particular species, that the number of hosts each species has chosen, that is ideal for them. And then there are a few, mainly parasitologists, who contend that 50% of the extent, or the living species at the moment, are all parasitic. Because if you think about it logically, it makes sense. Usually, every species will have at least one parasite species that infect it, usually more. And some parasites will be specific to that host. So if you have a 1-to-1 ratio of parasites to host, then you have 50% of the species of parasites. And within that area, there's just a vast range of numbers of multi-host, different combinations of host systems. But yeah, my personal parasites are anywhere between 1 and 4.
Melanie says, sorry if I missed this, but are the parasites you study invasive species, or are some of them local?
A little bit of both, actually. That's kind of exciting, because there's two species of invasive snail that have brought invasive parasites with them. There's one that has the only population in BC is in Crescent Beach. The parasite is called, the snail is Trittia obsoleta. It's a mud snail, and it brought 2 different species of parasite with it. It was moved, same kind of, I think it was the same vector where it was introduced with seed that was packaged around oysters. So Trittia obsoleta came from the eastern seaboard of North America, brought 2 parasite species with it, and then Batellaria atramentariae, that Japanese mud snail, came from Japan and brought 1 species of parasite with it. And the interesting thing is that if a snail arrives and it's inside it has that spore cyst that's creating the cercaria, the larvae, and then the larvae come out, if the snail's just arrived from the eastern seaboard, the cercaria is not going to have any idea what's going on, because the remaining host in its life cycle are suddenly 3,000 miles away in the opposite direction. So they are going to have to, on the fly, invent a new host system. So by definition, that means that some native BC crabs or birds will become infected by an invasive parasite. And then there's also the kind of exciting idea that the snail, or the parasite in the snail, will switch and start to infect a native snail. So then it'll essentially, that could be part of a process where it's becoming a new species. But for the most part, it's a mix. At this stage, it's also quite difficult to identify to the species level what parasite we're looking at, because you've seen they have quite simple body plans. That's quite common when you exist inside another organism. You don't need elaborate features, and the more simplified your structure, the better it is to absorb nutrients. So it's definitely an area that's absolutely fascinating in terms of describing new species.
Fantastic, thank you. Sheila asks, have you looked at other Litterina species for parasites as well?
Yes, I have looked at Litterina siccana. There are fewer infections in Litterina siccana, but that may actually be because of the way that I call it screening snails for parasites, which basically just means looking through a large number of snails to try and find the infected ones. And one of the common ways of doing that is you put the snails in about 3 milliliters of seawater, and you warm the seawater up to room temperature and shine a light on them, and that quite often triggers the release of the larval stages. So that's a brilliant non-destructive way of finding out whether the snail is infected or not. But some snail species, they don't respond to those stimuli as well as, so this actually happens. Litterina scuculata, quite often all the parasites will come out if you warm them up slightly. Litterina siccana doesn't do that, so usually you have to freeze the snails and then dissect them and then physically look, which is just a much slower process. Although that's part of the reason why I'm finding so many parasites at the moment, because I have a lot of time to sit in my house, dissect snails, and actually found that 15% of the Litterina scuculata at Spanish banks are infected, which is a much higher infection level than I was expecting. I've also looked at Litterina littorea, which is a common periwinkle from the Atlantic. There's been a couple of cases where people have found, and myself included, have found Litterina littorea in Burrard Inlet, but none of them have been infected with parasites so far. So that's kind of, we'll wait and see how that one goes.
Yeah, just imagine if we went into a sauna and parasites just started coming out of us. Okay, okay. So it's Sheila says we had a thank you from Kasia for an answer as well. But Sheila says, thinking of swimmer's itch, are these parasites apt to affect humans in any way? Hmm.
Only the one of the species that came was Trittia obsoleta from the eastern seaboard and took up residence in Crescent Beach. That is the only, and so swimmer's itch is caused by those larvae that come out of the snail. They bump into your leg while you're swimming and mistake you somehow for the next host in their life cycle. So they kind of pierce the outer skin and then cause this almost like an allergic reaction. That's quite a specific group of parasites, and I haven't come across any other ones other than in this one little pocket at Crescent Beach. But while I was in Crescent Beach collecting snails, I did get approached by a few members of the public saying that they were slightly unimpressed that this invasive snail had come with it and brought a parasite that causes swimmer's itch. I quite agree.
We had another thank you from Melanie, and also Kashifa asks, could you talk a bit more about the cysts? What purpose do they serve? Also, did you say they might be helpful to the host?
Let's see. So yes, that might have been one of the bits when I was talking too fast for my computer to keep up. So the cysts they're kind of almost like a seed. They're a resting stage. So the larva will come in and enter somehow the next host, whether it's a crab or a barnacle, and over maybe a week or 2 weeks, it will transform from the larvae into this sphere that I had in those photographs. And its function is really to just essentially live as long as possible. It's going to kind of enter something analogous to a dormant state, and the longer it stays viable inside that crab or barnacle, the more likely it will be eaten by the next host in its life cycle. So really, they just have that one purpose and one function, and they do have an effect on that host, but usually it's negative. There's a really interesting case from New Zealand where I did my PhD, where you have a bivalve, kind of like a clam, and the cysts form in the foot muscle of the clam. And so as a result, if you have enough cysts, it interferes with the muscle function of the foot muscle, and the clam can't bury itself as deep in the sand, which actually means that usually there's a little bit of it poking up above the level of the sand, and then that makes it more vulnerable to being eaten by a shorebird, which then completes the parasite's life cycle. It's just another example of the effect on those hosts is observable, but it is usually, unfortunately, negative for the host, right?
And then Jeffrey says, has anyone isolated and studied the DNA of these different species?
Yes, but in my personal opinion, not enough. Taxonomy, the sort of description of species that are out there is quite, it's a process that moves quite slowly. And recently I've become very interested in doing exactly that, preserving some tissue. We would then purify the DNA, we'd send it off to a company who would sequence that DNA and send us back a section that we would be able to compare with other sections of DNA that had been put into a kind of repository or a library. Now, that's a brilliant system. Unfortunately, it only works if somebody has already sequenced and analyzed the species that you're looking at. So really, I think we're kind of at the beginning stages of a really exciting period where we get to really identify what we're working with to a quite specific level, specific literal terms as well, a level. And the reason why I became excited about this is that sometimes there's this idea of cryptic biodiversity. And that basically just means you have a group of organisms that are absolutely identical. You think they're one species, but they turn out to be a sort of, they call it a cryptic lineage, or just a number of species that all look identical. And the one way that we can really get at teasing apart these different members of the cryptic lineage is by sequencing DNA and comparing them between two. I'll definitely say that there's another benefit to doing genetic work here. And I can make a kind of researcher's educated guess that the snail is infected by the parasite that then affects the crab. But if I was able to sample both the larvae coming out of the snail and the cysts from the crab, then if they match, then I am starting to really get a quantifiable and a firm idea of where those connections are and what this parasite species uses as its hosts. So yeah, that's an exciting area that I'd love to move into.
Mhm. That's great. I'm guessing that there aren't any Facebook live stream messages, but feel free to jump in if you see any. Oh, we got one more message from Sheila I'm going to read. Okay, I understand that some parasites are considered to co-evolve with their hosts. Do you think that this might be the case with the trematodes that you have studied?
I have a feeling that most parasites have co-evolved with their hosts. So I was talking earlier on about generalists versus specialists, and there are trade-offs between the two. So generalists don't rely on one particular species of host, which means that, again, they're kind of using probability. They develop the ability to infect a wide range of species. And those ones may be less likely to have co-evolved with hosts. And but the other side of that argument are highly specialized parasites, and potentially by specializing on a particular species of host, they get very good at circumventing the immune system, and then they can maybe reproduce more within that host. But the idea of this coevolution in parasitology meetings, we quite often call it a kind of arms race between host and parasite. So the host will develop or evolve some new ability to repel the parasite, and then the parasite will evolve some new ability to get around that recent defense mechanism. And that's essentially that is exactly what coevolution would be. So I think that because at least for some hosts in the trematode's life cycle, they are highly specific. I think it's likely that there is some element of host and parasite coevolution across the majority of trematode species.
Fantastic, thanks. Okay, so there's one more now. So Kashfa asks and says thank you as well. She says, Colin, do you have any other favorite host parasite systems? Perhaps ones that you got interested in studying parasites?
Yes.
So
so
origin story.
Yeah, the origin story actually, yes. So when I was doing my undergraduate degree at University of British Columbia, I did an honours thesis at the Vancouver Aquarium, and it was not on parasites at all. It was on beluga whales and the vocal development of a beluga calf that was born at Vancouver Aquarium. And it was interesting, but I was there's not an awful lot of experimental work in working with whales, because obviously I'm not going to let you do anything to them. And I talked to my supervisor at the time, and he actually said it was not just from an experimental perspective. He said that quite often, I mean, working with whales is a very competitive area. So why don't you try use maybe, he suggested toxicology or parasitology. And obviously he was suggesting parasites of cetaceans. And I was like, okay, that sounds brilliant. I'm definitely going to go for the parasitology one, because toxicology sounds a little bit too chemistry. I'm still kind of frightened of chemistry after organic chemistry in second year doing my undergrad. And I talked to my one of the professors at UBC. Do you know any host parasite systems that I could do a cool project on? And they recommended that there was a single-celled ciliate parasite that infects Physaster acraceus. So everybody, most people will probably have seen Physaster acraceus. There's these gorgeous purple or ochre sea stars. And some of them had become infected by this parasite, and the parasite took up residence in the gonads of male sea stars only. And I went out and collected sea stars and extracted some of these little ciliate parasites. And they were brilliant. You could look at them underneath the compound microscope, and they kind of looked like little mice. They were making decisions about which direction to go in. They feed on yeast fragments or bits of bacteria, so they're looking at very small food morsels. And this is during my undergrad, so I hadn't quite developed the repertoire of keeping parasites alive at the time. So I read a paper and said, okay, this yeast fragments, I'm going to get a solution of yeast fragments. So I used a solution of salt water and marmite. And it worked amazingly. I kept a culture of these things under my lab bench for ages, because I would just pull them out and look at them underneath the microscope when I was bored, because they just looked fascinating. And so that's my origin story. That's how I went from cetaceans to marine invertebrates, and I never looked back.
That's great. I wasn't expecting that. Okay. On behalf of everybody, Colin, we really want to thank you for sharing your time with us. And I was actually very impressed with the questions as well, right? And I think it was really lovely for you to do this for us. Thank you.
Oh,
And
you're very welcome.
I'm going to show a couple of just some slides just to sign us off. But thank you again. Slides.
Absolute pleasure.
Thank you. Okay, so just the museum. We have the questions. Just in case anyone's looking for a way to support the Beaty Biodiversity Museum in this time, and if you're able to, then these are a couple of links that you can follow. And the next section, we've got behind the scenes with Chris Stinson, Curatorial Assistant of Mammals, Reptiles, and Amphibians for the Cow and Tetrapod Collection. So that'll be next Wednesday, same day of the week, same time. And please do join us on social media at Beaty Museum. You can find a lot of online resources at this website here too, the Beaty at Home. And we'd love to hear your input about our online sessions too. This is brand new for us. We're still figuring things out. So please do fill out that Beaty survey as well. Thank you. Thanks everyone. And we're gonna sign off now and we'll see you next week, hopefully. Okay.